So imagine a muscle that can maintain this incredible vice-like grip for 24 hours straight.
Right. Burning almost zero energy.
Exactly, and just never getting tired. Like if the biceps in your arms tried to do this, they would just exhaust their cellular fuel completely.
They'd cramp up and basically fail in, what, a matter of minutes?
Yeah, a few minutes tops. Yet inside your body right now, you have millions of these tiny unsung muscles performing this exact miracle. I mean, they are regulating your blood pressure, they're digesting your last meal, and, they're silently keeping you alive.
It's incredible.
It really is. So today we are taking a deep dive into the microscopic mechanics of human physiology, specifically the excitation and contraction of smooth muscle.
Yes.
We are extracting the hard data, the chemical pathways, the voltage numbers straight from Chapter 8 of the text. And we're stripping away all that academic dryness to uncover the deep structural truth of how your body actually works and, well, why you should care.
And I'll be jumping in to translate all those technical points into real-life medical situations, clinical pearls.
Right.
Because it is genuinely fascinating when you realize that skeletal muscle, the biceps, the quads, the muscles that get all the glory in the gym, they only handle voluntary movement.
Yeah, they're the show horses.
Exactly. But smooth muscle is the actual workhorse. I mean, it operates completely beneath your conscious awareness. And to understand how it achieves these seemingly impossible feats of endurance, we have to look at the raw physical blueprints.
Which brings us to the first section. The text lays out the physical dimensions, and they are honestly almost unbelievably small.
Oh, totally microscopic.
Yeah, a single smooth muscle fiber is only about 1 to 5 micrometers in diameter, and maybe 20 to 500 micrometers in length.
Right.
To put that into perspective for you, a skeletal muscle fiber can be 30 times wider and hundreds of times longer. So we're talking about absolute microscopic threads here.
But despite being so small, they generate an incredible amount of force. And the way they are organized dictates exactly how they function in different parts of your body.
Right, the architecture dictates the capability.
Exactly. And the body primarily uses two entirely different architectural layouts for smooth muscle. You have multi-unit and you have unitary.
And they could not be more different, right?
They're completely different behaviors. So multi-unit smooth muscle is essentially a collection of independent, discrete fibers. Each individual fiber is totally separate.
It just operates on its own?
Yeah, on its own. And it's usually controlled by a single nerve ending. They're even insulated from each other by this tiny layer of collagen and glycoprotein basement membrane.
So they're fully physically isolated.
Precisely. They never touch.
So if they're all acting independently, where do we actually see that in action? Like, why would the body want a muscle to act as a bunch of isolated units instead of one big team?
It really comes down to the need for absolute microscopic precision. Think about what happens when an ophthalmologist shines a bright pen light right into your eye.
Oh, your pupil constricts instantly.
Right. It has to constrict instantly to protect the delicate retina from light damage. The iris of your eye is actually made of multi-unit smooth muscle.
Oh, wow.
Yeah. Because each fiber can contract independently, your nervous system can make these incredibly fine micro-adjustments to the exact diameter of your pupil.
Yeah.
You don't want a clumsy all-or-nothing contraction there.
Right. You want a dialed-in response.
Exactly. Another great example is your skin during a fever chill.
Oh, like getting goosebumps.
Yes. Your sympathetic nervous system fires, and these tiny independent piloerector muscles pull on individual hairs, making them stand on end and giving you those goosebumps.
That makes total sense. Precision and independence. But what about the other layout, the unitary smooth muscle?
Right. Unitary.
The textbook data says this is also called syncytial or visceral smooth muscle. And the term unitary is actually a bit of a trick, isn't it? It doesn't mean one single giant muscle fiber.
No, it's a massive misnomer. It trips up a lot of medical students.
I bet.
Unitary refers to a mass of hundreds or even thousands of individual smooth muscle fibers that contract together as a single coordinated unit.
Ah.
So instead of being isolated like the multi-unit fibers, these cells are arranged in large sheets or bundles. And this is the crucial part. Their cell membranes actually physically adhere to one another at multiple points.
So they're physically fused together.
Essentially, yes.
Yes.
Which means when one fiber pulls, it literally drags its neighbors along with it, transmitting mechanical force directly through the tissue.
But wait, I have to ask. Mechanically linking them is one thing, But how do they coordinate the actual command to contract?
Good question.
Because if you have hundreds of thousands of cells in a massive sheet-like in the walls of the intestines, how does an electrical nerve signal not just, fizzle out before it reaches the end?
Right, or cause a chaotic storm where half the tissue is contracting and the other half is relaxing.
Exactly. How
Yeah.
does it stay organized?
This is where we see one of the most elegant structures in the human body, the gap junction.
Gap junction.
Yeah. Gap junctions are essentially tiny protein tunnels connecting the insides of these adjoining cells. They act as high-speed electrical highways.
So the ions can just cross right over.
Instantly. Instead of relying on a nerve to individually shock every single cell, an electrical impulse hits one area, and the ions flow freely through these gap junctions to all the surrounding cells.
Wow.
The action potential washes over the entire organ simultaneously. This guarantees a unified, synchronous response.
Which is absolutely critical in a real medical scenario. Like, the text mentions childbirth.
Absolutely.
The uterus, which is this massive structure of unitary smooth muscle, it must contract as a single coordinated organ to generate the immense force required to push a baby through the birth canal.
Right. If the uterus were made of multi-unit muscle, you would just have millions of random, uncoordinated twitches. Nothing would move.
Yeah, that would be completely ineffective.
You see the exact same thing in the gastroenterology department. The wave-like peristaltic movements of your GI tract have to operate as a coordinated sheet to smoothly push a meal through your intestines.
So if those gap junctions failed?
If they were compromised, the signal would scatter. You'd get localized spasms that prevent the passage of food, leading to severe, agonizing bowel obstructions. The unitary design prevents that chaos.
Okay, so to lock this in for everyone listening, what's the mnemonic here?
Right. For the types of smooth muscle, remember MIP and UVGU.
MIP and UVGU.
Yeah. MIP stands for multi-unit equals iris and piloerector. And UVGU stands for unitary equals visceral, gut, and uterus.
MIP and UVGU. Perfect.
Yes.
So
Right.
let's get even smaller for a second and look inside these specific cells. The internal architecture here is totally alien compared to skeletal muscle.
Completely different.
Skeletal muscle has that classic striated, striped look because all the actin and myosin filaments are lined up in perfect, rigid rows called sarcomeres. But smooth muscle doesn't have those stripes at all.
All right, it throws the rigid sarcomere rulebook right out the window.
Instead, it has this wild geometric web. The actin filaments are attached to structures called dense bodies.
Dense bodies, yeah.
And some of these dense bodies are anchored right into the cell membrane, and others are just floating, suspended inside the cell. And the text notes that the membrane-dense bodies of adjacent cells actually bond together to form intercellular protein bridges.
Which is exactly how the physical force gets handed off from one cell to the next. And those dense bodies are basically doing the exact same job as the Z-discs in skeletal muscle.
They act as the anchor points.
Exactly. When the muscle contracts, it pulls those dense bodies closer together.
But the way they pull is what really blows my mind. So you have the myosin filaments sitting between the actin filaments.
Right.
The myosin is thick. The text says it's more than twice the diameter of the actin, but there is way less of it. There are like 5 to 10 times more actin filaments than myosin filaments.
A huge ratio difference.
Yeah. And the myosin heads, the little cross bridges that grab the actin and pull it, they are arranged in what is called a side-polar configuration.
This is such a brilliant piece of anatomical engineering.
Mm.
In skeletal muscle, all the myosin hinges are pulling the actin toward a center line.
Right.
It works, but it can only shorten the muscle by about 30% before the physical structures physically crash into each other.
They hit a wall.
Right. But the side polar arrangement in smooth muscle is entirely different. The myosin hinges on one side of the filament are pulling an actin filament in one direction,
Okay.
while the hinges on the exact opposite side of that same myosin filament are grabbing a different actin filament and pulling it the other way.
So they're actively pulling in opposite directions simultaneously?
Exactly. It's a back-to-back sliding mechanism.
Mm.
And because they are sliding past each other in opposite directions, there is nothing to crash into.
Oh, wow.
The muscle cell can just keep pulling and pulling until it wrings itself out, shrinking by an astonishing 80% of its original length.
An 80% contraction. That is a massive reduction in size. Why is that specific physical capability so necessary for human survival?
It's because of the specific types of organs smooth muscle has to operate, specifically hollow organs.
Like the bladder.
Exactly. Take the human bladder. When it fills with urine over several hours, it expands massively. When it's time to void, that muscle has to contract drastically to empty out completely.
Right.
If your bladder could only contract 30% of its length, like your bicep, it would never fully empty.
You always have urine left over?
Constant residual urine, which would lead to severe chronic bacterial infections, pressure building up the ureters, and eventual kidney failure.
That is terrifying.
It is. The same would apply to the stomach, right?
Yeah.
If your stomach could only shrink by 30%, you would develop severe gastroparesis.
Which is a dangerous blockage.
Right. The food just sits in the stomach and rots because the muscle literally cannot squeeze tightly enough to push it all into the small intestine. So that side polar hinge is the mechanical savior of our hollow organs.
That is fascinating. Do we have a mnemonic for this architectural section?
We do. Remember DAMS, dams. Dense bodies act as Z-discs. Actin dominates. Myosin has side polar bridges.
Dams. I love it. Okay, moving on to section 3 in the text. This brings us to the performance metrics of smooth muscle, which are, frankly, completely counterintuitive at first glance.
Oh, they don't make sense until you really look at the chemistry.
Right. We established that smooth muscle can contract down to 20% of its original size, but it does this incredibly slowly.
So slow.
The cycling of the myosin cross bridges, meaning the time it takes for the myosin head to grab the actin, pull, release, and reattach, is abysmally slow. The data shows it's 110 to 1300 the frequency of skeletal muscle.
And the biochemical reason for that sluggishness is that the myosin heads in smooth muscle have incredibly low ATPase activity.
Right, ATP being the energy currency of the cell.
Exactly. In skeletal muscle, the enzymes chew through ATP aggressively, firing off rapid-fire contractions. Smooth muscle just does not have that chemical appetite. It degrades ATP at a fraction of the speed.
Because of this, the energy required to sustain a contraction is practically a rounding error. Only 110 to 1300 as much ATP is needed.
It is the ultimate biological low-power mode. And it translates to a very slow overall physical movement.
Yeah, the text says contraction doesn't even begin until 50 to 100 milliseconds after the tissue is excited. It takes half a second to reach peak contraction, and then another 1 or 2 seconds to fully relax. A single contraction cycle can take anywhere from 1 to 30 full seconds.
Which is about 30 times as long as a single twitch in a skeletal muscle.
But here's the paradox that really stands out in the data. You have fewer myosin filaments. You have an incredibly slow cycling speed. You are burning barely any ATP.
So you'd think it's weak.
Exactly. You would assume this makes for a weak, flimsy muscle. Yet the maximum force of contraction is actually significantly greater in smooth muscle than in skeletal muscle.
Which is wild.
Right. Skeletal muscle tops out around 3 to 4 kilograms per square centimeter. Smooth muscle can generate 4 to 6 kilograms of pure force per square centimeter of cross-sectional area. How is it generating so much power with so little energy and speed?
It all comes down to leverage and time. Because the cycling is so slow, those cross bridges, the microscopic arms pulling the muscle tight, spend a much larger fraction of their time physically attached to the actin filaments.
Oh, so they aren't constantly letting go.
Right. They aren't resetting like in skeletal muscle. They just grab on and hold. And that persistent cumulative grip translates to immense sustained force.
And this data perfectly explains how our cardiovascular system survives.
Yes, exactly.
Your blood vessels, specifically the arterioles, are wrapped in smooth muscle. They have to maintain a state of continuous active contraction, what we call vascular tone, every single second of your entire life just to keep your blood pressure up.
If the smooth muscle in your arteries burned ATP at the same aggressive high-performance rate as the skeletal muscles in your legs do during a sprint, the metabolic demand would be catastrophic. The human body could not consume enough calories in a day to keep up. You would literally starve to death just from the basal metabolic energy required to prevent your blood vessels from relaxing and collapsing.
Wow.
By requiring up to 300 times less energy, your body can sustain critical, life-saving blood pressure 24/7 without exhausting its cellular fuel.
Let's get the mnemonic for this slow and powerful section.
Think SLOW. Slow cycling, low energy, outstanding force, widespread tonicity.
Slow, that's a good one. Now that endurance aspect leads straight into section 4, the latch mechanism.
The latch mechanism is brilliant.
The text describes this as a state where once the smooth muscle has reached its full contraction, the nervous system can drastically dial back the excitatory signal. But despite the signal fading, the muscle maintains its full crushing force of contraction.
Right.
And it does this using virtually no additional energy. It essentially locks into place.
And thank goodness it does, because the latch mechanism is what allows your gastrointestinal sphincters to do their job.
It's like in the stomach.
Exactly. Think about the lower esophageal sphincter, which sits between your esophagus and your stomach. It has to remain clamped completely shut for hours on end to prevent highly corrosive stomach acid from splashing up and destroying your esophageal lining.
Oh, heartburn.
Right, acid reflux. Because of the latch mechanism, that sphincter can maintain a watertight, high-tension seal all afternoon while you digest lunch, without tiring out the muscle tissue and without draining your body's systemic energy reserves.
It just latches the biological lock. But smooth muscle also has this other superpower when it comes to endurance called stress relaxation and reverse stress relaxation.
Yes.
This is the ability of unitary smooth muscle to return to almost its exact original resting tension just seconds or minutes after it has been massively stretched or shortened.
This is a property that actively saves your kidneys every single day.
Really?
Oh, absolutely. Let's go back to the urology patient whose bladder is slowly filling with, say, 300 milliliters of urine. As the fluid first enters the bladder, it physically stretches the smooth muscle wall. Now, if you stretch a rubber band, the tension spikes immediately. And initially, the internal pressure in the bladder does spike.
That makes sense.
But here is the danger. If that pressure stayed high, the urine would literally be forced backward, up the ureters, and into the delicate filtering structures of the kidneys.
Oh, causing renal damage.
Devastating, irreversible renal damage. But that doesn't happen because of stress relaxation. Within 15 to 60 seconds of that initial stretch, the smooth muscle actively rearranges its cross bridges.
It just adapts.
Exactly. It elongates and relaxes just enough for the internal pressure to drop right back down to normal, safe baseline levels. The bladder seamlessly accommodates the new volume without maintaining dangerous pressures.
That is incredible engineering.
And when you finally void, reverse stress relaxation happens. The bladder shrinks down, but the tension quickly normalizes so the organ doesn't collapse into a rigid spasm.
It constantly adapts its internal tension to its physical volume automatically. So, mnemonic for this one.
Latch. L-A-T-C-H. Less ATP, tension continues high.
Less ATP, tension continues high. Perfect. But let's dig into the chemistry of how it actually pulls the trigger to start contracting. This is section 5.
The chemical pathway.
Right. This brings us to a major fundamental difference between smooth and skeletal muscle. Skeletal muscle relies on a regulatory protein called troponin to initiate contraction. But smooth muscle doesn't have a single drop of troponin.
It just doesn't exist here.
Which means the entire chemical cascade to start a contraction has to operate on a completely different pathway.
And understanding this specific pathway is arguably one of the most important things a medical student will ever learn, because it is the exact target of some of the most prescribed drugs in the entire world.
Okay, let's unpack that pathway step by step from the text.
Yep.
It all starts with calcium. Step 1. The concentration of calcium ions inside the cytosolic fluid of the smooth muscle cell rises.
Right.
Step 2. These newly arrived calcium ions bind reversibly with a large regulatory protein called calmodulin.
Calmodulin, yeah.
Step 3. This newly formed calcium-calmodulin complex joins forces with and activates an enzyme called myosin light chain kinase, or MLCK.
Let's pause on MLCK for a second. Kinases are enzymes that slap a phosphate group onto things. That is their whole functional job.
Right, which leads perfectly to step 4. The activated MLCK phosphorylates or slaps a phosphate onto the regulatory light chain on the myosin head.
Exactly.
And finally, step 5. Once that regulatory chain has that phosphate attached, the myosin head is activated. The attachment-detachment cycling with the actin filament finally begins, and the muscle physically contracts. It is a very deliberate 5-step domino effect.
And this exact chemical sequence is manipulated by doctors millions of times a day.
How so?
Let's look at a patient suffering from dangerous hypertension, high blood pressure. Their vascular smooth muscle is clamped down too tight. A physician will often prescribe a calcium channel blocker, like amlodipine.
Oh, I've heard of amlodipine.
Right, it's incredibly common. This drug is elegantly designed to physically intercept that exact 5-step pathway we just discussed. Amlodipine circulates in the blood and physically blocks the microscopic channels that allow calcium to enter the smooth muscle cells of the blood vessels.
So it completely stops step 1.
Exactly. By preventing the calcium from entering the cell, the drug stops the dominoes from falling. Without an intracellular calcium spike, step 2 fails. There is no calcium to bind to calmodulin.
So the complex never forms.
Right. Therefore, the MLCK enzyme is never activated, the myosin head is never phosphorylated, and the smooth muscle is chemically incapable of contracting.
Wow.
As a result, the blood vessels relax, they dilate, The vascular resistance drops, and the patient's blood pressure falls to a safe level, potentially saving them from a catastrophic stroke. Understanding that calmodulin pathway is not just textbook trivia, it is the fundamental mechanism of saving lives.
That is a brilliant application of the chemistry. Mnemonic for the pathway.
CCMP, calcium binds calmodulin, activates MLCK, drives phosphorylation.
CCMP. Got it. But wait, let me push back on this pathway for a second based on what you just said.
Sure.
We established that phosphorylation, adding the phosphate, is the definitive trigger that starts the contraction. So, logically, does a simple dephosphorylation immediately stop it? Like, is it just a light switch you can flick off to cause instant relaxation?
That is exactly what you would assume.
Yeah.
Right. But the reality is much more complex and deliberate. Reversing the contraction isn't just throwing the engine into reverse. It requires active chemical intervention.
Which brings us to section 6.
Right. The fascinating problem of where the calcium comes from and how the cell eventually gets rid of it to relax.
Let's dive into that. Finding the calcium. In skeletal muscle, virtually all the calcium needed for contraction is stored perfectly inside the cell, locked up in a vast network called the sarcoplasmic reticulum, or SR.
Right, the SR.
When the muscle gets shocked, the SR dumps the calcium and boom, contraction, but smooth muscle. The text points out that the SR in most smooth the muscle cells is barely developed. It's rudimentary.
Which presents a massive physiological hurdle. If the cell doesn't store its own calcium internally, it has to look outside.
Precisely. The vast majority of the calcium that drives smooth muscle contraction has to flow into the cell from the extracellular fluid, ECF, out in the bloodstream and interstitial spaces.
Right.
The text notes the concentration of calcium out there is huge greater than 10 to the minus 3 molar compared to less than 10 to the minus 7 molar inside the resting cell. There is a massive gradient just waiting to rush in.
A huge difference in concentration.
Yeah, but because the calcium literally has to diffuse through the membrane from the outside world, it takes time. This causes a latent period of 200 to 300 milliseconds before the contraction even begins.
And, 200 to 300 milliseconds sounds fast to a human, but in cellular biology, that is an absolute eternity.
The text explicitly says it is 50 times longer than the latent period for skeletal muscle.
Exactly.
Now, to help manage this, smooth muscle does have a small structural adaptation. The cell membrane features these tiny pouch-like invaginations called caveolae.
Caveolae.
Yeah. These caveolae sit right up against the meager sarcoplasmic tubules inside the cell. When an action potential sweeps over the membrane, it dips into these caveolae, which seems to excite the little bit of SR that is there to release whatever tiny calcium stores it has.
And the text suggests these caveolae are basically a primitive, rudimentary version of the highly advanced T-tubule system you find in skeletal muscle.
Right. But the critical takeaway here is that heavy, absolute reliance on the extracellular fluid for the bulk of the calcium.
Which creates a severe clinical vulnerability. Let's look at a patient presenting in the emergency room with severe hypocalcemia.
Meaning low calcium.
Exactly. Their blood calcium levels have dropped to dangerously low levels due to kidney disease, parathyroid issues, or severe malnutrition. Because their vascular smooth muscle relies entirely on that external calcium gradient to initiate contraction.
Yeah.
A drop in blood calcium severely diminishes the ability of the arteries to maintain vascular tone.
The smooth muscle literally runs out of raw chemical fuel.
Exactly. It wants to clamp down, but the calcium simply isn't there to enter the cell and bind to the calmodulin. This can lead to a drastic, life-threatening collapse in the patient's blood pressure.
That's terrifying.
And notice that skeletal muscle doesn't suffer this exact same immediate catastrophic failure during mild hypocalcemia because it has its own private reserve of calcium safely locked away in its well-developed sarcoplasmic reticulum.
Oh, wow.
Smooth muscle is at the complete mercy of the environment around it.
That perfectly illustrates why the ECF reliance is such a big deal. So, returning to my relaxation question, how does the cell clear all that calcium out to stop the contraction?
Right, to turn the switch off.
The text explains it requires an active, energy-consuming process. The cell uses a slow-acting calcium pump that physically forces the calcium back out into the ECF or pumps it into the small SR. This pump requires ATP to function.
Which is an interesting paradox. You actually have to burn energy to relax the muscle.
Right. But pumping the calcium out isn't enough on its own. The myosin head still has that phosphate attached to it.
Yes.
So relaxation also strictly requires an enzyme located in the cytosol called myosin phosphatase. This enzyme's sole job is to actively hunt down the myosin heads and chemically split the phosphate off the regulatory light chain. Only when that phosphate is removed does the cycling finally stop.
And here is where we get the biochemical explanation for that amazing latch mechanism we talked about
Yeah.
earlier.
How so?
Think about what happens when the calcium levels start to drop. The kinase enzyme, which puts the phosphate on, deactivates. But if the myosin phosphatase enzyme, which takes the phosphate off, is also deactivated or acting very slowly, the myosin heads remain phosphorylated for a much longer period of time.
So they are stuck in the on position.
Precisely. They maintain the static latching force without needing to constantly cycle and degrade new ATP. The enzymes basically pause the biochemical clock, locking the muscle tight. It is an incredibly elegant holding pattern.
It really is. So what's our mnemonic for this calcium section?
Remember ECF and PP?
ECF and PP.
Yeah. ECF stands for extracellular calcium fuels contraction, and PP stands for pump and phosphatase for relaxation.
Awesome. Now moving on to section 7, let's shift from the internal chemistry to how the nervous system actually delivers the command. This is the wiring, the autonomic neuromuscular junctions.
All right, the wiring.
Skeletal muscle is famously controlled by these highly structured, perfectly engineered contact points called motor end plates. A nerve fiber comes down, plugs directly into a specific spot on the muscle fiber, and delivers a massive payload of neurotransmitters to guarantee a contraction.
It is a hardwired point-to-point connection.
But in most smooth muscle, those motor end plates completely do not exist.
Nope.
Instead, the autonomic nerve fibers branch out diffusely and just kind of lay on top of a sheet of muscle fibers.
Yeah.
The text calls these diffuse junctions. They don't make direct hardwired contact. Instead, they secrete their neurotransmitters into the matrix coating of the smooth muscle, sometimes from several nanometers or even micrometers away. The transmitter then just diffuses through the fluid to reach the cells. And the structural anatomy of the nerve fiber itself is different. Instead of one terminal button at the end of the nerve, the axons feature multiple bulging areas along their length called varicosities.
Varicosities, yes.
The text describes these varicosities as areas where the Schwann cells, which normally insulate the nerve, are interrupted. Inside these varicosities are hundreds of tiny vesicles packed with neurotransmitters. As the electrical signal travels down the nerve, these varicosities release transmitters all along the length of the nerve fiber at once.
Now, you have to note the exception here.
Right.
While the vast majority of unitary smooth muscle uses this diffuse volume transmission, multi-unit smooth muscle like the iris in the eye we discussed requires much faster, more precise control.
Right, for the micro adjustments?
In those specific multi-unit fibers, the text points out that the varicosities are separated from the muscle cell membrane by a tiny gap of only 20 to 30 nanometers. These are called contact junctions, and they allow the multi-unit fibers to react with lightning speed compared to the slow, wave-like diffusion required for a whole sheet of visceral gut muscle.
Okay, so the structural wiring varies, but what chemicals are actually being released out of these varicosities?
Right, the transmitters.
The text identifies two primary neurotransmitters for smooth muscle, acetylcholine and norepinephrine, and it lays down two absolute unbreakable rules regarding them. First rule, acetylcholine and norepinephrine are never, ever secreted by the same nerve fiber.
Never.
A fiber is either cholinergic, releasing acetylcholine, or adrenergic, releasing norepinephrine, never both.
And that prevents chemical crosstalk and confusion.
Right. But the second rule is the one that is truly wild to me. Acetylcholine might act as an excitatory transmitter that causes a heavy contraction in one organ, but act as a powerful inhibitory transmitter that causes complete relaxation in a different organ. And the same goes for norepinephrine. When acetylcholine excites a fiber, norepinephrine ordinarily inhibits it, and vice versa. The text states unequivocally that the action of the neurotransmitter does not depend on the chemical itself. It depends entirely on the specific receptor proteins located on the surface of the smooth muscle cell membrane.
This is so important.
The muscle dictates the response, not the nerve.
This concept of receptor-dependent action is the absolute cornerstone of emergency medicine and critical care pharmacology. It explains how a single drug can perform multiple miraculous tasks simultaneously.
Okay, give us an example.
Let's look at a terrifying but common scenario.
Yeah.
A patient is brought into the ER suffering from severe anaphylactic shock, a massive systemic allergic reaction to a bee sting or a peanut.
Oh, anaphylaxis.
Right. Their physiology is collapsing. Their airway smooth muscle is swelling and spasming shut, meaning they cannot breathe. Simultaneously, their vascular smooth muscle is rapidly dilating and relaxing, causing their blood pressure to crash towards zero. They're minutes away from death.
So the physician immediately administers an injection of epinephrine and EpiPen.
Exactly. Epinephrine is closely related to norepinephrine. That injection floods the patient's systemic bloodstream with this single chemical compound. It washes over every smooth muscle in the body at the exact same time.
Okay.
Now, if the chemical dictated the action, every muscle would do the exact same thing, which would be disastrous. But because smooth muscle response is dictated by the local receptors, this single molecule performs two life-saving, completely opposite actions at the exact same moment.
How does that actually play out in the lungs versus the blood vessels?
In the smooth muscles surrounding the patient's bronchial airways in the lungs, the cell membranes are packed with specific inhibitory receptors, beta-2 adrenergic receptors.
Inhibitory.
Right. When the epinephrine binds to these inhibitory receptors, it rapidly forces the airway smooth muscle to relax. The spasming stops, the airways open wide, and the patient can suddenly pull oxygen into their lungs again. But simultaneously, down in the blood vessels, the smooth muscle cell membranes are packed with specific excitatory receptors, alpha-1 adrenergic receptors. When that exact same epinephrine molecule binds to these excitatory receptors, it forces the vessels to constrict powerfully.
Which raises the blood pressure.
Exactly. It clamps down on the circulatory system and violently raises the crashing blood pressure back up to a normal perfusing level. One single chemical transmitter. Two completely different, highly targeted physiological actions dictated entirely by the microscopic receptor proteins built into the membrane of the smooth muscle.
That perfectly illustrates why the receptor rule is so critical. The body is essentially outsourcing the decision-making to the local tissue rather than relying on the central nervous system to send two different perfectly timed signals.
Exactly. Oh, and the mnemonic for the wiring is VAN. Van varicosities carry acetylcholine or norepinephrine.
Love it. Which brings us to Section 8. The electrical signals themselves, the action potentials.
The electrical spark.
We know that the normal resting membrane potential of smooth muscle hovers around -50 to -60 millivolts. For context, that is about 30 millivolts less negative than skeletal muscle, meaning smooth muscle sits a little closer to the threshold, a little more ready to fire.
And when it does fire, unitary smooth muscle produces action potentials in two totally distinct flavors. The spike potential and the action potential with a plateau.
Let's define the spike potential first. The text notes, these are pretty straightforward. They look a lot like the action potentials in skeletal muscle. They shoot up rapidly, they repolarize quickly, and the whole electrical event is over in 10 to 50 milliseconds.
Very fast.
They can be triggered by nerves, hormones, or stretch. But the second flavor, the action potential with a plateau, is highly specialized. The text explains that the plateau action potential starts with a normal rapid electrical spike, but instead of dropping right back down to reset, the repolarization phase is artificially delayed.
Delayed for a long time.
Yeah, the electrical charge stays positive for hundreds of milliseconds, sometimes up to a full 1,000 milliseconds, a whole second.
And that electrical plateau translates directly to a prolonged mechanical contraction.
Exactly. But chemically, mechanistically, why does that plateau happen? Why doesn't it just reset immediately?
It's the channels.
Right, the text points directly to the membrane channels. Smooth muscle cell membranes are structurally unique. They are packed with far more voltage-gated calcium channels than skeletal muscle, but they contain very few voltage-gated sodium channels.
Right.
In skeletal muscle, fast sodium channels pop open, sodium rushes in to cause the spike, and then they slam shut instantly. But in smooth muscle, calcium flow is primarily responsible for generating the action potential.
And calcium channels operate on a completely different timetable.
They do. Calcium channels open sluggishly, and more importantly, they stay open much, much longer. Because they remain open, the positive calcium ions continue to flood into the cell, keeping the voltage high and creating that massive electrical plateau.
Right.
But the text also highlights a brilliant dual function here. The calcium entering the cell isn't just an electrical charge carrying a message. That exact same calcium physically binds to calmodulin to activate the MLCK pathway we talked about.
Yes.
It uniquely performs two tasks at once. It generates the electrical action potential, and it provides the physical chemical spark that acts directly on the contractile mechanism to cause the roll.
Which is incredibly efficient biology.
Yeah.
And if we look closely at that plateau action potential in a clinical setting, we can go right back to the obstetrics ward to see why it evolved this way.
What with the uterus?
Exactly.
Yeah.
We talked about the uterus needing to contract synchronously.
Yeah.
But it also needs to contract for a long duration. During childbirth, If the uterine smooth muscle only fired off rapid spike potentials, the muscle would simply twitch briefly and reset.
Just a quick twitch.
Right. And a brief, half-second twitch is physically incapable of advancing a baby through the birth canal. The uterus must generate an immense crushing force and sustain that force for several seconds to be effective.
So the plateau is essential.
The plateau action potential provides the exact electrical foundation for that. The slow calcium channels keep the cell depolarized, which keeps the cross bridges cycling, ensuring the smooth muscle contraction is prolonged and sustained.
The physiological electrical data perfectly matches the mechanical requirement of the organ. That is amazing. Mnemonic.
SAP and CC. SAP stands for spike and plateau. CC stands for calcium conducts and contracts.
SAP and CC. Got it. Now let's look at section 9, another unique electrical feature of smooth muscle. The pacemakers.
Pacemakers, yes.
The text covers slow waves and stretch responses here, and it makes a very firm, explicit distinction right away. Slow waves are not action potentials.
They are not.
They do not cause muscle contraction on their own. Instead, they're a basic underlying rhythm of the membrane potential. The voltage just slowly waxes and wanes, drifting up and down by a few millivolts.
Think of it like a continuous underlying electrical hum. It's just a baseline fluctuation.
The text suggests these slow waves might be caused by the waxing and waning of the pumping of positive ions, presumably the sodium pump pushing ions outward through the membrane, or by rhythmic changes in ion channel conductances. We don't know for absolutely sure.
Right.
But what we do know is their vital function regarding the threshold. The resting potential is around negative 60 millivolts. The text explains that the threshold to trigger an action potential is about negative 35 millivolts.
Okay.
When the peak of one of these underlying slow waves drifts high enough to cross that negative 35 millivolt threshold, boom! An action potential develops. It spreads over the muscle mass via the gap junctions and the gut contracts. This is why they are called pacemaker waves. They set a baseline rhythm that eventually elicits rhythmical contractions.
And this pacemaker activity is massively amplified by physical stretch.
Yes, excitation by stretch. When visceral, unitary, smooth muscle is stretched sufficiently by an object inside it, it generates spontaneous action potentials. Mechanistically, this happens because physically stretching the cell membrane pulls it, making it leakier to positive ions. This decreases the overall negativity of the membrane potential. So if the membrane naturally sits at negative 60, stretching it might drag it up to negative 45.
Moves it closer to the threshold.
Exactly. When you combine that new, less negative baseline with the peaks of the natural flow waves we just discussed, the voltage effortlessly hits that negative 35 millivolt threshold. The slow wave crests, the threshold is breached, and the action potentials fire in a rapid sequence.
This stretch excitation mechanism is the entire basis for human digestion.
How so?
Let's consider a patient who has just sat down and eaten a massive, heavy holiday meal. As that tremendous volume of food leaves the stomach and begins entering the intestines, it physically distends and overfills the gut wall.
Stretching the tissue.
Right. The simple mechanical stretch of the tissue physically pulls on those membrane channels, lowering the resting membrane potential. The tissue becomes less negative. Combined with those continuous underlying pacemaker slow waves, the membrane potential repeatedly breaches the threshold.
And fires the AP.
This triggers automatic, rhythmic, and incredibly powerful peristaltic waves. The smooth muscle behind the food distends, contracts, and propels the food forward toward the anus.
And what is wild is that you don't even have to think about it.
Exactly. From a medical and evolutionary standpoint, this is brilliant. This entire massive, coordinated digestive effort requires zero conscious input from the central nervous system. You don't have to consciously command your gut to digest.
The stretch does the work.
The localized mechanical stretch of the food itself is enough to change the electrical voltage of the cells and trigger the entire digestion process. The organ is entirely self-sufficient.
Mnemonic for this one.
SSS, stretch, stimulates spikes.
SSS. Perfect. Which brings us perfectly to our final textual segment, section 10, and honestly the most mind-blowing fact in the entire chapter.
Really is.
Going action potential free. We just spent 20 minutes dissecting action potentials, thresholds, and calcium channels, and then the text drops this staggering statistic. Approximately half of all smooth muscle contraction in the human body is initiated without action potentials at all.
Half. That is a staggering figure, especially considering how much emphasis medical science places on the electrical action potential in baseline physiology.
Right.
To realize that 50% of the time, The body completely ignores the electrical action potential system and uses alternative pathways.
Hmm.
It's incredible.
It really makes you rethink how the body is controlled. The text details how this action potential-free contraction works in 3 distinct ways. First, in multi-unit smooth muscle, the fibers are simply too microscopically small to generate a self-propagating action potential.
They're just too tiny.
Right. Instead, when the neurotransmitter hits the membrane, it causes a localized depolarization called a junctional potential. Because the fiber is so tiny, this junctional potential simply spreads electrotonically over the entire fiber.
And, spreading electrotonically can sound like dense jargon, but it just means the electrical charge disperses naturally without needing a new current to push it.
Just a natural diffusion of charge.
Exactly. The muscle fiber is so small that a single local charge is enough to trigger the calcium influx for the whole cell.
Okay, the second way smooth muscle operates without action potentials involves local tissue chemical factors. And this heavily applies to the tiny blood vessels, the arterioles, and precapillary sphincters.
Very important clinically.
These vessels have little to no nervous innervation. They are controlled purely by the chemical environment immediately surrounding them.
The local factors.
The text lists the specific local chemical factors that cause these smooth muscle sphincters to relax and cause massive vasodilation. A lack of oxygen in the local tissue, an excess of carbon dioxide, increased hydrogen ion concentration, the presence of adenosine, lactic acid, increased potassium ions, nitric oxide, increased body temperature, and decreased blood pressure.
That's a big list.
If any of these factors build up in the tissue fluid, the smooth muscle physically relaxes, opening the blood vessels wide without a single nerve firing.
This localized chemical control is the ultimate evolutionary survival mechanism.
Give me an example.
Let's look at a patient in the emergency department suffering from severe tissue ischemia, say, a lack of oxygen in a limb because a major artery is partially blocked. The cells in that leg are suffocating.
Right, they need oxygen immediately.
They are rapidly burning through their remaining oxygen, and as they struggle to survive, they are dumping massive amounts of carbon dioxide and lactic acid into the surrounding tissue fluid.
The waste products of anaerobic metabolism.
Right. Now, if the blood vessels in that dying leg had to wait for a signal from the brain to dilate and bring more blood, the tissue might die. But because of local chemical factors, the collapsing oxygen levels and the rapidly building lactic acid act directly on the smooth muscle of the precapillary sphincters,
Oh, bypassing the brain?
completely bypassing the central nervous system. The acid and the low oxygen physically force the smooth muscle to relax and open as wide as mechanically possible. This attempts to pull every possible drop of available blood flow into the dying tissue. The local tissue is literally attempting to save its own life, based purely on its own chemical waste products.
The autonomy is just wild. Now, the third and final non-electrical pathway the text covers involves hormonal mechanisms and second messengers.
Second messengers.
Hormones circulating in the blood like norepinephrine, epinephrine, angiotensin II, endothelin, oxytocin, and histamine can bind to receptors and either directly open calcium channels to cause contraction or strongly inhibit contraction, all without generating an action potential. But the second messenger systems are the really complex ones. A hormone or a local factor binds to a receptor on the outside of the cell membrane. But instead of opening an ion channel, that receptor reaches inside the cell and activates an internal enzyme, usually adenylate cyclase or guanylate cyclase.
Right, those are the key enzymes.
These enzymes then construct the second messengers, cyclic AMP EC-CAMP or cyclic GMP CGMP. And these second messengers act as the internal signaling molecules. They roam around inside the cell, phosphorylating various enzymes that activate the calcium pumps. They actively command the cell to pump calcium out of the intracellular fluid, clearing out the sarcoplasm and forcefully driving the muscle into deep relaxation.
And the clinical pharmacology regarding these specific second messengers is arguably one of the most famous and widely used treatments in all of cardiac medicine.
Oh, really? Which one?
Consider an elderly patient who experiences sudden, crushing chest pain angina. They are having a lack of blood flow to their heart muscle, a potential heart attack. They immediately reach into their pocket and put a tiny nitroglycerin tablet under their tongue.
Ugh.
Nitroglycerin, a classic life-saving maneuver. But how does it actually work at the cellular level?
It relies entirely on the second messenger system. When the patient absorbs that nitroglycerin, the drug rapidly breaks down and releases nitric oxide directly into the bloodstream. That nitric oxide diffuses into the smooth muscle cells lining the coronary arteries of the heart.
Okay.
The nitric oxide directly and aggressively boosts the production of cGMP, the exact second messenger you just mentioned.
So the inside of the cell is suddenly flooded with cGMP molecules.
Precisely. And this
Ah.
massive army of cGMP aggressively activates the calcium pumps. They immediately sweep all the calcium out of the intracellular fluid. And as we learned earlier, without intracellular calcium, the calmodulin cannot activate MLCK, the myosin cannot phosphorylate, and the muscle physically cannot contract.
It has to relax. The smooth muscle in the coronary arteries rapidly relaxes and dilates, immediately restoring massive, life-saving blood flow to the suffocating heart muscle. The chest pain vanishes. The direct, undeniable line from manipulating a microscopic second messenger to resolving a lethal heart attack is one of the greatest triumphs of medical physiology.
That is just incredible. Mnemonic for this final section.
LOCAL and MAGIC. Local and magic.
Local and
The
magic.
local stands for low oxygen, causes arterioles to loosen. Magic stands for messengers adenylyl guanylate inhibit contraction.
Excellent. That is an incredible synthesis of everything we have talked about today. We have traced the journey of smooth muscle from its microscopic physical dimensions, establishing how multi-unit fibers give us precision, while unitary fibers use gap junctions to operate as massive coordinated sheets. We broke down the unique side-polar back-to-back actin-pulling architecture that allows hollow organs to shrink by 80% without destroying themselves. We unpacked the paradox of their incredibly slow, low-energy, but highly forceful mechanics that maintain baseline blood pressure without starving us.
And the latch mechanisms that lock sphincters tight for hours.
Yes. We tracked the precise calcium-calmodulin chemical pathway, analyzed the risky reliance on the extracellular fluid for calcium, and explored the diffusion via autonomic varicosities. Finally, we explored the plateau action potentials of the uterus, the self-starting stretch responses of the gut, and the massive impacts of action potential-free local tissue factors and second messengers saving tissues from ischemia.
It is a massive amount of intricate biological data. But as we step back and connect all of this to the bigger picture of where medical science is heading, the sheer, staggering autonomy of smooth muscle is what truly stands out.
Yeah, autonomy is the perfect word for it.
Think about what we just discussed. This tissue is constantly thinking for itself on a microscopic local level. It reacts instantaneously to a slight drop in oxygen. It mechanically adjusts to the physical stretch of an overfilled bladder. It intercepts circulating hormones, all while perfectly coordinating these local adjustments to keep the entire organism alive without you ever having to consciously direct it.
It is a completely decentralized intelligence system running our bodies.
It really is. And this raises an incredibly provocative question for the future of medicine.
Okay, lay it on us.
If approximately half of all smooth muscle contraction is governed by local tissue chemical factors and internal second messengers rather than electrical nerve impulses from the brain, what does the future of targeted pharmacology look like?
That's a great point.
Historically, we have treated systemic diseases by throwing drugs at the entire central nervous system, hoping to force a change and accepting all the massive systemic side effects that come with it. But as bioengineering advances, will we eventually abandon the nervous system entirely when treating things like hypertension or gastrointestinal disorders?
Just skip the brain entirely?
Imagine a future where we focus exclusively on bioengineering hyperspecific synthetic second messengers, customized KMP and cGMP modifiers, that only activate in the presence of specific local chemical signatures. We could potentially control diseases exactly where they happen, at the exact local tissue level, manipulating the muscle cell from the inside out without causing a single systemic side effect in the rest of the body.
Treating the cell at the source, bypassing the brain entirely. That is a remarkable mechanism and a truly fascinating future to consider. Thank you for joining us on this meticulously detailed medical exploration into the microscopic world of smooth muscle physiology. We hope this deep dive has given you the precise, real-world understanding of how your body works beneath the surface. Keep questioning, keep learning, and we will see you on the next deep dive.